I've seen more smiling faces today than I've expected. That's good. What are your thoughts? The thoughts, whenever I ask thoughts usually are like this. Kind of a nonverbal thought. Nope. Feedback? Perspectives? Was it too easy? It wasn't too easy? So I should make it harder next time? Yes, ma'am.
The long answer questions, it felt really, really specific.
Uh-huh.
Compared to, like, there were questions that
Yeah.
were so specific, I felt like it could have been a short answer question.
Uh-huh. Yeah. My long answer questions like those will tend to be specific when they ask for several things. Yeah, you're right, they are. And part of the reason for doing that is I find that actually students feel a little more comfortable if they know exactly what I'm asking them. But if I say, tell me about translation or transcription, then it's like, oh my God, what am I going to do? You know, and then they start writing everything and anything and everything, hoping they get something with it. So that's part of the reason for doing that. But I understand what you're saying, yeah. Other thoughts? Yeah?
I wish that you did more short answers or like essays in the first lesson because sometimes I feel like the points are really distributed.
Okay. So you like more short answer questions worth fewer points? Is that what I understand?
I mean, like, I would rather have 5 small paragraph answers that are 5 points rather than 3 that are 3 paragraphs for 15 points.
Yeah, I'll tell you, when I do that, and I appreciate the feedback, but when I do that, what I run into is people start running out of time. Because if I make more, and I've tried this, if I try to do more than 3 long answers of any sort, everybody, oh, I can't get your exam done. And it's like, oh. So, yeah, but I understand what you're, I mean, I think personally, the more questions you have worth fewer points, the more territory you can cover. And I certainly sympathize with that. But one of the things in this class that's different, and I teach BB 450. I teach BB 450, 451, and 350. And on the BB 450 class, they take a somewhat similar format exam as you guys do. And on the first exam, I try to get as many questions as I can so that, you know, for what you said. And that's where I run into trouble. And you know, that class has 400 students, and they'll all come and go, oh, my God, it's just too much. And it's like, okay, cut back. Yeah?
Have you thought of maybe like reducing the points that they're worth because like the other questions are like 3 points and then the longer questions would be worth more, or sometimes like if you miss one.
Yeah, yeah. Yeah, and that's also a good question and another consideration. One of the reasons for making them worth more points, of course, is that there's in theory, more in them. But I will take your suggestion of more short answer questions on the next exam. I'll do that. So, how many got complimentary right? You haven't? Nobody's looked? You haven't looked? Yeah. It's COMPLEMENTARY. So. Okay. All right. Well, let's move forward. We have some territory to cover. I plowed through a lot of translation last time, and so I've gotten through the mechanism, the general mechanism of translation. And if we look at translation and compare prokaryotic and eukaryotic, the overall process is basically the same. The names of the proteins and all the various things in eukaryotic are different. We don't care about that. That's the process that matters. And the process, if you understand the process for one, you understand the general process for the others. And I've pointed out some of the differences as I've gone along. I'll point out a couple more differences today. But they're not as great as you might think. Okay? One of the places where they're different enough is in the response to certain chemicals. So as you can imagine, proteins, of course, have very specific structures. And when we look at a ribosome, we have dozens of proteins that are in there. So it's not totally surprising that those proteins that are in ribosomes might be targets for drugs, and they are. Okay? And there are enough differences in the structure of the proteins in the prokaryotic ribosome compared to the eukaryotic ribosome that a drug can specifically be designed to knock out the function of a prokaryotic ribosome but not affect a eukaryotic ribosome. And that turns out to be a really powerful thing. So anytime you have a compound that only works on one class of organisms, then you've got a very specific kind of antibiotic. And so I've got a couple of examples up here, but there are drugs that specifically target proteins or processes in prokaryotic translation that are different than eukaryotic. And if you think about it, if you were to knock out translation for an organism, you're going to kill the organism because the organism has got to make proteins in order to divide, to be alive, to do the things that organisms do. All right. One of those drugs that targets a process in ribosomes is this compound called puromycin. And you don't need to worry about the structure, but if you look at the structure, what you'll see is that it at least partly resembles the end of a tRNA. And that end of a tRNA can actually make it into a ribosome. If it makes it into the ribosome, what will happen is the ribosome will accept it, but then it can't process it. It can't do anything with it because it doesn't have the right structure as an actual tRNA molecule does. So what this puromycin will do is it will actually get attached onto the end of a growing polypeptide, okay? But then it's stuck. And so the ribosome can't process, the ribosome can't go any further, and everything just stops. So you're stopping the process right in the middle of making a protein. And that's actually how puromycin can kill cells. Other antibiotics that target processes in ribosomes include chloramphenicol. Chloramphenicol is an antibiotic that will specifically hit the ribosome of prokaryotic cells and knock out prokaryotic cells. Tetracycline will also do that. Okay? So, all right. If we turn our attention to eukaryotes, I've already talked about a significant difference in the structure of eukaryotic messenger RNAs compared to prokaryotic messenger RNAs. So I'm going to remind you that eukaryotic RNAs underwent a considerable amount of modification after they were made. First, at the 5' end, there's a cap that's put onto there, and that's that unusual nucleotide structure that I told you about earlier. Second, at the 3' end, there's what's called a poly-A tail that's put on, and that's just a string of dozens and dozens and dozens of A residues. I was a little hurried when I talked about this last time, so I'm going to mention it to you here now because I think it's an important and interesting phenomenon. These two modifications that happen to eukaryotic messenger RNAs actually have a couple of functions. One of them, as we'll see in a minute, is that it helps the translation process to proceed efficiently. But another important consideration for these is that these modifications of the ends of the messenger RNA help to stabilize the messenger RNAs. What does that mean? Well, cells, if you think about it, make RNAs, and cells also break down RNAs. They have enzymes in them called RNases, R-N-A-S-E. An RNase is an enzyme that you can think of for RNA like we talked about a nuclease for DNA, or an exonuclease for DNA. These RNases are abundant inside of cells. And they are there because cells, once they've made a messenger RNA, they don't want that messenger RNA being around forever. We've talked about how it would be inefficient, for example, to be making the proteins necessary for lactose metabolism if lactose isn't there. So that means that not only does the cell want to control when it synthesizes that messenger RNA, but it also wants to be able to break down that messenger RNA so it's not floating around and being translated into protein when the cell doesn't want that protein. So cells are full of RNases. Okay? If you ever work with RNA in a laboratory, you'll find that RNases are some of the worst things that you'll ever encounter because RNases are everywhere. If you touch anything, your hand is so full of RNases that it will degrade any RNA you're trying to work with. Okay? And not only that, but the RNases are very stable. Okay? I talked about RNase earlier in the term being a very stable protein that we could heat up and it would renature. That is, it would come back to being active. And so RNases are very problematic that way. Well, one of the things that helps to stabilize a messenger RNA in a eukaryotic cell is the cap and the poly A. The cap actually prevents exonucleases from starting at the 5' end and chewing inwards. It physically prevents that from happening. So the 5' end of eukaryotic messenger RNAs is protected by the cap. At the 3' end, one of the reasons that cells put hundreds of A residues in is because the RNases that start chewing in on a cell are exonucleases. They start at the end and move inwards. And the longer that track of A's is, the longer it takes the RNases to degrade that tail. They will degrade a few bases and they will fall off. And they will degrade and they will fall off. So the longer that tail is, the longer that messenger RNA will be around. So both the cap and the poly A tail on a messenger RNA help the messenger RNA to be stable for a longer period of time. We can imagine there might be some messenger RNAs in a cell that the cell would want to keep for a longer period of time, and some messenger RNAs in a cell that it wouldn't want to keep for a longer period of time, and it wouldn't have as long of a poly A tail on it. And that turns out to be the case. Okay. Well, so we've got a cap, we've got a poly A tail. And another thing that I want to say about this actually relates to translation. Okay? Oh, sorry, got the wrong thing here. Okay. This depicts the translation process that occurs in a eukaryotic cell. Okay? The translation process. What you see in green is the messenger RNA, and the 5' end with the cap is right here at where my pointer is, where it says 7mg. Okay? The 3' end goes all the way up around here. What you see is an initiation complex, and we talked about initiation complexes in the initiation phase of translation before. In eukaryotic cells, they're more complex, and that initiation starts with two things. One is a protein that binds to the cap, and the other is a protein that binds to the tail. And I don't even care if you know the names of those proteins. That's not what matters here. Okay? So you've got a protein that binds to the poly A tail, you've got a protein that binds to the cap, and then you have several proteins, and you understand all the IFs there. Remember, IF stands for initiation factor. Okay? You see various initiation factors that are binding to each other and between there to make this structure a sort of a circle. And it turns out that in eukaryotic translation, that circle is thought to play a role in keeping the ribosome close to the initiation codon. What does that mean? Well, it means that as the ribosome starts here where the AUG would be, it translates, it gets through there, and it's over here, and it hits the stop codon. It's not very far away from the AUG again. Whereas if that end is not held close, as it is in this case, then the ribosome would have less of a chance of coming back to the AUG and starting another round of translation. So this circle is thought to facilitate that efficiency of the translation process. Now, I mentioned when I talked about prokaryotic translation that prokaryotes have a sequence called the Shine-Delgarno sequence, which is located a few bases ahead of the initiation codon. And I said it played a role of helping to position the transfer RNA, the messenger RNA, at the proper place in the ribosome so that the AUG was in that P site, remember? But I said that the eukaryotic cells do not have a Shine-Delgarno sequence. And you might wonder, how is it then that the ribosome gets positioned properly on a eukaryotic messenger RNA? And I'm going to partly answer that question for you as best we know it. Okay? The partial answer to that question is if we examine the sequences of messenger RNAs in eukaryotes, the first AUG that starts from the 5' end is usually the place where translation starts, meaning it's not the second or the third or the fourth or the fifth one. In prokaryotic cells, that's not the case. Prokaryotic cells, they need to have some signal that says this is the AUG to start translation from, because the first one won't necessarily be where the translation is supposed to start. So in this case, what the ribosome can do is it can start assembling anywhere in here, and then it can slide down until it hits the first AUG and says, here's where we start putting in methionine, and we start the translation process. A prokaryotic cell can't do that because the first AUG isn't necessarily where translation is supposed to start. Okay. Questions about that? You guys are too worn out from the exam, I can tell. Okay. So that's the eukaryotic process. Elongation in eukaryotes continues very much like elongation in prokaryotes. There is the bringing in of the tRNAs like you saw. There is the covering up of that bond between the tRNA and the amino acid by that protein, that EFTU-like protein that carries in the tRNAs. And there is a protein like EFG that does the translocation step. And yes, GTP is the energy of translation in eukaryotic cells as well. So a lot of similarities to the prokaryotic process. Same thing happens when we get to termination. There's some minor differences in termination, but overall the process is basically the same. A stop codon appears in the A site. A release factor comes in and basically pulls everything apart, and translation stops, and the peptide is released, just like we saw in the prokaryotic cell. Okay, so that's the translation process that I want to talk about for both prokaryotic and eukaryotic cells. I want to talk for a few minutes about things that happen after a protein has been made. Okay? Things that happen after a protein has been made. What these modifications are they're called post-translational modifications. And you've already seen some post-translational modifications. For example, converting a zymogen into an active protein involved cutting a peptide bond. And that was a post-translational modification. You've also seen adding a phosphate or taking phosphates off of a protein, and those are also post-translational modifications. Post-translational modifications are very important for getting a protein into its final form to be functional. And one of the best examples of this is the protein that we know as insulin. Insulin is what we call a peptide hormone. So we'll talk a little bit more later about how insulin is a hormone. But it's a peptide hormone because it's a protein. And the protein is made originally as a fairly long chain, or a relatively long chain of amino acids. And the initial thing that's made in the synthesis of insulin is a protein called pre-pro-insulin. Pre-pro-insulin, okay? Excuse me. And you can see that pre-pro-insulin looks like this, and it's got some sulfhydryls that are the side chains of cysteine amino acids. And those side chains can fold, or the polypeptide can fold, and those side chains can be brought into close proximity such that they react and form disulfide bonds like you can see right here. That formation of the disulfide bonds is absolutely critical because in the further processing of insulin, what we see is that a couple of peptide bonds are broken. We see one broken here, we see one broken here. And when that happens, this guy that started out as one long piece now is really two short chains, and the only things that hold them together are those disulfide bonds. Okay? Insulin in the pre-pro form doesn't have any function, but insulin in this form over here on the right is essential for our body to be able to handle glucose. Okay? It's essential for our body to be able to handle glucose. Glucose, as I will talk about in a little bit, is something that is essentially a poison to our body. It's essentially a poison. We'll see why that's the case. And insulin helps the body to deal with the poisonous effects of glucose. So insulin is a very important protein, and it's made in a longer form and then processed down into a smaller form that you can see here. Okay. I've talked about how it is that RNAs can get broken down by RNases. And I've talked about how exonucleases can break down DNAs to some extent. Proteins also get broken down inside of cells. And you might think cells are they're making things and they turn around and break them down. You know, why do they do these things? And they do them because damage over time can occur. In the case of DNA, we saw replication damage, or we saw chemical damage that could happen that meant that parts of the DNA had to be removed and replaced. In the case of RNAs, we can have either damage occurring or having an RNA floating around that we don't want floating around. And we can also imagine that with proteins, we could either have chemical damage that occurs to a protein to destroy its function, or we could have a protein that, hey, we don't want that function around right now, we're going to break that protein down. Okay? Well, cells have structures excuse me, within them whose job it is to break down proteins. And how do they know which ones to break down? Well, it turns out that there's a flag that's put onto proteins that are destined to be broken down. Okay? There's a flag that's put onto them, and this actually shows one of the flags. The most common flag that's on there is a small peptide called ubiquitin. Ubiquitin attachment to a protein flags that protein for destruction. And one of the things that will destroy a protein is a structure called a proteasome. Proteasome. A proteasome is basically a complex that breaks down proteins into smaller pieces. And those smaller pieces can be further degraded into making something that is useful for the amino acids that are in it. Okay? All right. Let's see. So that's what proteasome does. Another interesting consideration in proteins, and this is not a post-translational modification, but this is actually a very interesting and very odd amino acid that is actually put into proteins as they're being made in some cases. It's an amino acid that we refer to as the 21st amino acid. Well, we always talk about how there are 20 amino acids that are found in proteins. So what's a 21st amino acid mean? Well, it means that when people have examined proteins over the years, they would occasionally come across this amino acid that's known as selenocysteine. It's called selenocysteine because that Se right there is the atom known as selenium. Okay? Selenocysteine looks just like cysteine, except for it has a selenium in place of the sulfur. Okay? This amino acid can actually be attached to some tRNAs and can be incorporated into a growing polypeptide chain. Now that means it's not post-translational modification. It's a translation that's happening that puts this into a protein. Okay? The mechanism by which that happens is beyond what we're going to talk about in this class. But you should know that selenocysteine can be incorporated during the translation process. And there are several proteins in our bodies and in virtually every cell that contain some selenocysteine. So where we talk about there being 20 amino acids, there really are 21. And this one gets in by an odd mechanism. As I said, I'm not going to talk about it here. But there is a way to get this translated into a protein. Okay. The last thing I'm going to talk about is something I mentioned briefly earlier. Yes, question? Yeah, what does selenocysteine do that's different from the other amino acids? Because it's got that selenium ion in there, we could imagine that the chemistry would be a little bit different and would allow for different kinds of reactions to be catalyzed or catalyzed in different ways. And that's actually what it does. Yeah. It's not a very satisfying answer, but that's what the selenium part of that actually does. And it's actually for that reason why selenium is an essential trace nutrient. You have to be very careful with selenium, though, because selenium, at anything above trace amounts, is poisonous. So you don't just go out and gobble a bunch of selenium. Okay. I always worry about that when I tell people, oh, well, yeah, here's this great nutrient. And everybody goes, oh, wow, we're going to have a whole bunch of that. Not good. Okay. The last thing I mentioned earlier in the term, and I want to mention it here again because it's appropriate, and that is a protein complex called a chaperone. And a chaperone is important to help a protein to fold properly. It helps a protein to fold properly. And what it provides is a chamber within which that protein can fold without interacting with other outside things. Okay? So a chaperone is important to help a protein to fold properly. Not all proteins need chaperones. But some proteins do need chaperones in order to fold properly. You'll see up here that there's a sort of a complex of this, and this complex has something called HSP70. HSP stands for heat shock protein. And when cells get heat shocked, they will usually induce the synthesis of chaperones. What does heat do to proteins? It unfolds them. What do chaperones do? They help to properly fold them. So heat shock proteins are important in helping, some of the heat shock proteins are important in helping other proteins to fold properly. Okay, and the two that we'll talk about, in fact, in terms of name, these are found in E. coli, the Gro-El, Gro-Es. And it's called the Gro-El, Gro-Es complex. And you can see the Gro-Es is basically a cap, whereas the Gro-El is the chamber within which that protein is inserted and is allowed to properly fold. After it is folded properly, it exits, and everything is done. Okay. That's the last of what I want to say here about translation. I want to turn our attention now to the use of our knowledge of proteins and DNA and RNA to make things. Okay? To make things. So we hear about the term genetic engineering. All right? There's a big ballot measure in Corvallis right now about genetic engineering, if you guys are aware of that or not. And people get very uptight with that term, genetic engineering, but in fact, genetic engineering is something that has occurred in nature since day one. We are getting the tools that nature uses to do genetic engineering. That's what we're doing. And because of these tools, we're able to do some pretty incredible things. And I'll give you a couple of examples. Okay? Some of the most remarkable products, some of the most remarkable medicines that are available today only because we have genetic engineering. Prime example, hemophiliacs. Okay? People who have hemophilia are lacking a protein or proteins that allow their blood to clot properly. In the days before human genetic engineering of proteins, the only way we could treat people who had hemophilia was either to take thousands of gallons of human blood and purify out tiny microscopic quantities of the clotting factor that those people lacked, and then inject it back into them so they didn't bleed to death every time they cut their finger. Well, that was fine and dandy until we had a blood supply that got contaminated with HIV. And there were dozens or hundreds of people who were hemophiliacs who died because the blood supply was contaminated that their blood clotting factors came from. Okay? When they, and this all came about the same time as the genetic engineering of this happened. Because of the fact that we could take and isolate from human cells the gene that codes for the missing clotting factor, or factors, we could put those genes into bacteria, have bacteria make those clotting factors. HIV was never present. Nothing else was very present. And not only could we make tiny amounts, we could make tons of it. Cost went down, quality went up, and people lived much normal lives as a consequence of that. That's one example. There are hundreds of examples that exist today. Okay? Human growth hormone, one good example. Another good example is one that just escaped my mind. Blood clotting, the anti-clotting factors. So there's a protein called tissue plasminogen activator, TPA, that dissolves clots, that has revolutionized the treatment of certain heart attacks and stroke issues where blood clotting was involved. You can actually inject this into a person and have their clot be dissolved almost instantaneously, which is really remarkable. Okay, so biotechnology has a lot of practical applications. Okay? The practical applications are, I want to talk a little bit about how we do all of those things, and hopefully show you in the process that we're not really playing God, as people like to describe that. We're not playing God. Okay. Well, we've talked a little bit about separation, and I'll remind you what you're looking at on the screen is something that we've already talked about, gel electrophoresis. And specifically, this is agarose gel electrophoresis, and specifically what you see on the screen is the separation of DNA fragments. The separation of DNA fragments is a critical process to be able to analyze and to be able to isolate specific DNAs that are of interest to us. Okay? How do we get fragments of DNA? Well, there's a variety of ways that we can get fragments of DNA, but one of the most remarkable of these was something that was discovered back in the 1970s, and that was that there were enzymes that were found in bacteria that would cut DNA at specific sequences. They would recognize a specific sequence, they would bind to it, and they would cut the DNA at that point. These enzymes are called restriction enzymes. They're also called restriction endonucleases, but I'll call them restriction enzymes here. Okay? Restriction enzymes. And these restriction enzymes are found throughout the bacterial kingdom. You look in a bacterial cell, in all likelihood, it's going to have some kind of a restriction enzyme. So I want to tell you a little bit about how they work, what they do, and how they're really useful to us for biotechnology purposes. Well, actually, I'll start with that. We can imagine that if we take a human chromosome that's several feet long, and we want some part of it that's a few microns in size, we would like to have a way to precisely cut and to obtain that fragment of DNA that we're interested in. Well, to do that, that means we have to be able to cut DNA, and we have to be able to cut DNA in places that we would desire. And so one of the ways we do things is by cutting that DNA with restriction enzymes. These restriction enzymes can be purified from bacteria, mixed with the DNA, and when they're mixed with the DNA, they will find their target sequence, bind to it, and then make cuts like what you'll see on the screen. So here's an example of one restriction enzyme called EcoR1. It's one of the most well-known restriction enzymes in the world. It recognizes the sequence GAATTC. So I'm only showing a part of a long DNA strand there, and in the middle of it is this sequence GAATTC. If you look at GAATTC, you'll notice that if you read the top strand from the 5' to 3' direction, it reads GAATTC. If you go to the bottom right of the bottom strand, you'll see reading left to right from 5' to 3', it also reads GAATTC. It's something that molecular biologists mistakenly call a palindrome. It's not a palindrome. technically, but it's a molecular biological palindrome. The top strand reads the same as the bottom strand. Most restriction enzymes recognize palindromic sequences. And that turns out to be useful because the enzyme has evolved the ability to cut, in this case, between a G and an A within that sequence. By recognizing a palindromic sequence, it can cut between this G and A, and it can cut between this G and A. And when it does that, you make a double-stranded cut in the DNA. This type of cut that this is made is called a staggered cut, meaning that it's got some bases overhanging, as you can see there. And that actually helps if we decide to put the pieces back together. Put the pieces back together like a puzzle. Those pieces will fit if we put them back together in the proper way. And I'll talk about that in a bit. Well, actually, you can see it on here. Okay, so here's those pieces. We see that T compared with A, T compared with A compared with T, A compared with T, and we put those back together. In this case, we started with this one. We started with this one. There's no surprise it's going to go back together. But what if I took a different DNA that had also been cut with EcoR1, that had the same overhang, and I put it back with this one here? That is, I had one DNA here and a different DNA over here. This might be from an E. coli cell. This might be from a dog cell. If I did that and I put it together with DNA ligase, and here's where that DNA ligase that you've learned about is important because it puts pieces back together. If I do that, I have just made what we call a recombinant DNA. And it just simply means we've recombined two things together that weren't together to start. Now that might seem, again, there's this playing God thing, and there's all this concern that's out there about, I shouldn't say all this concern. Some people have it, a lot of people don't have the concern. But some people are very concerned about the fact that you make recombinant DNAs. Again, nature has been making recombinant DNAs for billions of years longer than human beings have been on the face of the earth. So this process is happening and has been happening long before humans were ever here. We're using it to put things together to make products. All right? We can make human insulin. We can make human growth hormone. We can make any human protein that's needed by using these techniques that I'm describing to you. Okay, so restriction endonucleases are important, and I started to tell you why they were interesting, and I got distracted because I'm an old guy and I get distracted. Okay? They're important, they're really interesting. The restriction endonucleases are in bacteria not just to give us something to play with, but they actually play a role in bacteria to help protect them against viruses. How does a virus work? Well, we're going to talk about viruses probably on Friday, but viruses work by attaching to a cell, and viruses have a protein coat on the outside that grabs a hold of a cell, and once they've gotten a hold of their target cell, they can inject nucleic acid into the cell that codes for some proteins that the virus wants. If nothing else happens, then that virus will go in, it will replicate, and it most likely will destroy the cell. Viruses are known for every cell on the face of the earth. Bacteria get viruses just like we get viruses. Okay? Well, in our case, we have an immune system that's protecting us against the virus. We might catch a cold, but we don't die from a cold because our immune system catches up, destroys the virus, and we get over the cold, and everything is fine and dandy. A bacterial cell doesn't have an immune system because a bacterial cell is a single cell. How does a bacterium protect itself against a virus? Well, the way that it does it is with a restriction endonuclease. Imagine that that cell is bound by a virus. The virus injects its DNA into the cell, and the cell has a restriction endonuclease that can cut the nucleic acid of the virus. You would kill the virus in the process. And that's why bacteria have restriction enzymes. Okay? They're protection against invading DNAs. Well, if you think about that, you might ask the question, well, how does it keep from cutting its own DNA? Because its own DNA is going to have these sequences as well. These sequences are fairly common. This sequence here will occur on average about once every 4,000 nucleotides. How does the virus keep from cutting up its own DNA. Well, in virus, how do bacteria keep from cutting up its own DNA? The bacteria, in addition to having restriction endonucleases, also have something called a modification system. Okay? So the restriction endonuclease is part of the system. The other part of the system is another enzyme. So the restriction enzyme recognizes the sequence and cuts it. The modification enzyme is an enzyme called a methylase, M-E-T-H-Y-L-A-S-E. Every time a bacterial cell has a restriction endonuclease, it also has to have a corresponding methylase. What does the methylase do? The methylase recognizes exactly the same sequence as the restriction enzyme. So the methylase recognizes GAATTC. But instead of cutting that sequence, what the methylase does is it puts methyl groups on that sequence. It's putting methyls onto the bases of that sequence. So what? Who cares? Well, who cares is the cell. Because the restriction enzyme will not cut if that sequence has been methylated. So the bacterium can put methyl groups on that sequence in its own DNA. Its own DNA will not get cut by the enzyme. But when this bacterial DNA comes floating into the cell, this doesn't have methyl groups on it, restriction enzyme cuts it, the virus is dead. You might say, well, what if the methylase, can it put a methyl group on the bacterial DNA? And the answer is, yes, it can. This always gives people worry because they want nature to be perfect. And nature is not perfect, but nature is pretty good. Usually the restriction enzyme gets there first. Usually the virus gets broken down. But because viruses exist, we know it doesn't happen every time, right? Sometimes the methylase wins. If the methylase wins, then the viral DNA is protected, and the virus is probably going to kill the cell. Make sense? So restriction modification systems are very important systems for protecting bacteria. We don't have restriction enzymes in our cells. We don't have them in our cells. Bacteria have them. We don't have them. But we also have an immune system, right? So we have a different setup for protection than what bacteria do. Okay. There's the methylation. There's the modification. There's target sequence, protection, etc. Okay. This shows a variety of restriction enzymes, and no, you don't need to memorize these sequences, but I show you that there are different enzymes that recognize different sequences. Some of them recognize staggered sequences, like EcoR1, there's that GA and there's that GA, so that we have overhangs. BAMH1 cuts between G and G and between G and G, and it leaves staggered overhangs. Some only recognize 4-base sequences, like HEY3, and it cuts between G and C, and it cuts between G and C. And if you look at this one, you say, it doesn't leave a staggered cut. It leaves what we call a blunt end, and that's fine too. So there's all kinds of examples about how these cut. Here's one called NOT1 that recognizes an 8-base sequence. Okay? And so there's a variety of things that different cells have. The naming of these comes from the name of the bacteria that they come from. So EcoR1 comes from E. coli, actually, that's found in your gut. Okay. Well, I've already talked about a way to make a recombinant DNA. This figure hopefully brings it to life a little bit. You can see how it is that we have a DNA that we can put another piece of DNA into. And it also introduces a term that I haven't talked about, but that it's a very important term for us in biotechnology. And this term is something called a plasmid. You've probably heard the term before. I want to make sure you know what a plasmid is. So plasmid is a small circular DNA that replicates in bacteria. It's a small circular DNA that replicates in bacteria. The size of a bacterial DNA is about anywhere from 1 million up to about 10 million base pairs. The size of a plasmid is typically on the order of a few thousand. So it's much, much smaller than the bacterial DNA, but it can replicate, and it can replicate in the same cell as the bacterial DNA. Well, plasmids turn out to be really useful for us because the larger a DNA is, the harder it is to handle without breaking it. If we try to handle, meaning manipulate in a test tube, a bacterial DNA, it'll break before we do anything to it. It's just too fragile. Plasmids, because they're smaller, are not so easily torn. That's what happens with the big ones. And as a consequence of this, plasmids are what we prefer to work with when we want to introduce a new DNA into a bacterium. Now, I'll talk about that next time. But what I want to show you here is how we make that DNA to put into a bacterium. Okay? Here's a plasmid. It's a circle. If I want to put a DNA into there, I've got to put a break in that plasmid to put in the foreign DNA. If the plasmid that I'm working with has a single site for EcoR1, for example, I can cut that plasmid with EcoR1 and I'll generate a structure just like you see right here. It's got a gap. Well, it doesn't stay like this. The ends flop around and everything. We're showing it in a circular form to give you an idea about what it looks like. But in fact, the ends are anywhere there. If we take that circle and we combine it or mix it with a foreign DNA that's been cut with the same enzyme, so let's say I took human DNA and I cut it with EcoR1. So now I've got EcoR1 cut human DNA and I've got EcoR1 cut plasmid DNA. If I mix them together, and I'm very careful about how I do it, and I put some DNA ligase in there, I will have made a recombinant DNA. Okay? That recombinant DNA I could put into a bacterium. And that bacterium will replicate that and make useful things for me if I'm clever about what I'm doing with it or how I designed it. Okay? So I've just made a recombinant DNA. All right. Now, I'm going to jump ahead and show you something that's a modified plasmid. Plasmids occur in nature. They're not man's inventions. But we have manipulated some of those plasmids to be useful for us. So the last thing I'm going to tell you about today is how we've manipulated those to use them. What does it take to make RNA? Do cells just automatically make RNA? What do they need? They need an RNA polymerase, but what does the RNA polymerase need? What? Sigma factor for what purpose? Needs a template, but need to have a promoter, right? So if I put a DNA into a plasmid and that plasmid doesn't have a promoter that RNA polymerase can recognize, I'm not going to be able to make any protein corresponding to that gene I've just put in there, right? So one of the things I need if I want to make proteins in a bacterial cell is I need to have a promoter. So an ideal plasmid is called an expression vector. What you see on the screen is an expression vector. The first of those factors that's needed is a promoter. Okay? Second of the factors that's needed is we have to have a replication origin. Well, since it replicates, we know it's there, but of course we have to have a replication origin. Those two things are critical. Now, I'm not going to talk about the last critical thing until tomorrow. I think we've gone through enough already today. But I do have a relevant song for you to sing, and it's about translation. It's fairly easy to sing. It's called Good Protein Synthesis, and it's to the tune of Good King Wenceslas. Amino acids cannot join by themselves together. They require ribosomes to create the tether. All the protein chains get made according to instruction, carried by mRNA in peptide bond construction. Small subunit starts it all with initiation, pairing up to RNAs at the docking station. Shine-Delgarno's complement in the 16 S's lines the AUG up so synthesis commences. Elongation happens in ribosomic insides, where our RNA creates bonds for polypeptides. These depart the ribosome, passing right straight through it in the tiny channels there of the large subunit. Finally, when the sequence of one of the stop codons parts itself in the A site, synthesis can't go on. P side RNA lets go of what it was holding, so the polypeptide can get on with its folding. All right, guys, see you Friday. Hi, how's it going?